this Q3 characterization, besides reverse engineering, is also called Q1, Q2. When you have to do reverse engineering in a formulation, means you are evaluating what kind of excipients are there and what is the quantitative sameness of the excipient. So that is again evaluation of Q1, Q2 sameness. So as I told you, Dissolution, in vitro permeation studies for oral dosage form, that also we started because just doing by IVRT, maybe our penetration could not be that good. So we thought could be some new technique which can attract the client attention. So we came up with Dissolution because there was a need in the market. Besides this, we also came up with another new aspect called microdialysis based IVRT studies. So this is microdialysis as a tool for in vitro release is indeed something which is very high on industrial priority, and people are evaluating it a lot as a in vitro release tool, because regulatory authorities have a lot of expectations when it comes to characterization of formulations like complex ophthalmics, and the conventional methods are not meeting fully on those kind of expectations. So people are evaluating microdialysis as a novel tool to address those expectations. So when we started in 2019, we formed the company. We form all the, basically when you start a company, you have to do a lot of filing work, registering the company, and then you have to do all kind of a paperwork to form a company, so that you have a regulated kind of a framework. And then we started approaching our clients. Since we had a good network, we had spent say 18 to 28 years of all our partners they had spent in the industry. So we had a good client base, or we had a good networking. So we started approaching the right person who knew the problem, who knew that there is a problem and there can be a solution if the path is right. So by virtue of our consistent efforts and by approaching the right kind of a people in the industry, we were able to achieve more than 200% growth in our first 2 years of operation. Parallelly, we also knew that we have to be an innovation-based company, so we have to be innovative. So we also worked parallelly in collaboration with the academic institutes to work on innovation front. We worked along with the GFSU, Gujarat Forensic Science University, on an innovative formulation project on respiratory liposome formulation under PACE program. We got Ibrt funding for respiratory liposome formulation. So that was, I think, very good achievement, because a number of applicants were many, for the institutes like IIT and various institutes. There was tough competition, but still we managed that funding. We got ISO 9001-2015 certification as a first step. And then I talked about microdialysis, since there was a lot of interest in the industry. But what we realized is whatever has been technique, there was very limited information in the literature, because these things are knowledge based and people don't reveal much in the public domain because they know the commercial potential of these. So generally, a lot of information doesn't come into public domain unless things get patented and all those things. So we realized whatever information was available in the domain, there are significant gaps with respect to technology, because it has to be robust, it has to be industrially friendly, QC friendly kind of a thing. So the technique reported in the domain was not QC friendly. So we came up with a novel microdialysis technique for which we filed PCT application, and so our technique was offering solution to the limitations which was there in the public domain. So that industry, we got very good response from the industry on our innovative microdialysis technique. Because why we got with good response? Because industry was facing challenges from regulatory agencies. They were consistently asking them tough questions about use of IVI technique for complex formulation. Like for ophthalmic formulation, you cannot evaluate in vitro release by using conventional type 1 or type 2 kind of a methodology, because the physiology is quite different in the eye. So you need to be innovative when you show that in vitro release evaluation of this kind of a formulation. So this is a microdialysis technique for which now we have a PCT patent application filed in global market. So that is something which is very important or very credit worthy for our organization. Then novel in vitro permeation, Disoplus that I mentioned, that is also, since these two were our USP, unique selling property. So basically these gave us good reputation in the industry. People knew that they are doing something different. Besides this, we also did some protein binding studies for cyclodextrin based complex injectables. That was again a challenging project that we got, and we performed it successfully. Then for complex carbohydrate, iron carbohydrate complex, there are certain regulatory requirements which are quite challenging. So we came up with an easy method for in vitro hemodialysis. That is again a proprietary method. So by virtue of all those small innovation, we established ourselves as one-stop solution provider globally. So now, if you talk about our client base, we have more than 40 clients, including various national, multinational companies, and all from regulated market, catering to US and Europe market. So that is something which is testimony to our success in the field. Our client base will tell, okay, the kind of success that we have achieved in our past 2 years, and I hope we will continue on the same path. So another to share another feather in the cap, like we are nomination. Our nomination is as one of the finalists for Startup India Award. So we are the finalist of this award by Government of India, and we are expecting a result in January 2022, and we are hopeful of coming up with flying colors in this competition. And based on our performance, last 2 years performance, we were in the position to attract more funding, because in our past facility we were small, only 1,000 square feet facility. Later on, which moved to say 2,000 square feet, we took another floor also, but still we were small. So there was scope for further growth. So we needed more funding. So then we were looking for right kind of a partner who can work. Basically, we were looking for strategic partnership. We were not looking for person who can just fund our venture. So in Sortex, we found the right kind of a partner, because they were Sortex, USP type 4 world leader in the manufacturing of pharmaceutical characterization machinery, like USP type 4, type 1, type 2. They are the world leader. This is a Switzerland-based company. So we are also into Ibrt, and Sortex is also having this fully GMP lab in SPS France, and they have also one more testing lab in US. So basically our ideology and basically our strengths were complementary. So we both talked to each other, and we thought, yeah, it makes sense to join hands, because our strengths are complementary and we can be benefited from each other. So that's how this new venture, this Sortex, came up around 6 months back. By virtue of this venture, now we have a 10,000 square feet facility near Changodar, and now this is state-of-the-art GMP analytical facility. So from research base, now we are moving to this GMP, because when you talk about GMP, a lot of cost is involved having that kind of, because regulatory requirements are quite stringent. And when we talk about GMP, it is basically to cater to the need of regulated market like US and Europe. So to meet those kind of expectations, this is a huge cost involvement. So in a nutshell, we strengthen our infrastructural and technical capabilities. And of course, since Sortex has a global client base, so we got reached to the global market also. Earlier, our clients were more from India or Asia Pacific market, but now with this joint venture with Sortex, we got global reach, and hopefully we shall reap the benefit of this joint venture in our new lab, which is just qualification and all things are going on. Another 1 or 2 months, it will be fully operational. Now with this new kind of a expansion, now we have our scientific advisory panel also to like Ms. Jayashri Sharma is again biopharmaceutical expert. So with the Dissolution kind of a, because you know when you talk about each one has to be subject matter experts. So for this Dissolution related biopharmaceutical evaluation of formulation, so she's quite good. Samir Hirochi is managing director of Stotex SPS France. So he's on our board, and he is world-renowned speaker when it comes to IVRT studies. So we are fortunate to have in our board and as one of the advisors. So any interesting project, we interact on daily basis and something for client-related needs. So basically, you know, when two people meet, cross-fertilization of ideas, there is some issue. So basically, all our knowledge, it combines to give solution to the need of the client. Okay, so now with the coming up with new venture, now we have strengthened our services. So in our new brochure, you will find various new services. Like now, reverse engineering and deformulation services we are offering with now we are having facility equipped with ion chromatography, GPC, right, ELSD detector, LCMS. So these with new kind of infrastructure, now we are able to RI detector. So we are able to meet reverse engineering characterization or deformulation need of our various clients. So again, Q3 characterization, when you say, I already mentioned what is Q3 characterization for IVRT and other. With respect to other kind of a Q3 characterization, we have planned to further strengthen our infrastructure in the coming year. So we offer one-stop in vitro release testing for using all type of USP apparatus, USP type 1 to 7. There is no other CRO in India which offers such kind of a dedicated services. Besides this, in vitro permission testing of topical dosage form. That is something, you know, that is really upcoming. When you in vitro release testing using French cell is quite common, people do it. But in vitro permission testing is one of the regulatory requirement in US, wherein you evaluate the skin permeation, human skin permeation, to prove the bioequivalence of the topical formulation. So this is kind of a testing that is very specific need for regulated market and which has a very huge cost implication. So we have, there are not many CROs in India performing this kind of a services. So we have started this, and we have acquired required infrastructure for the same. Again, Dissolution for oral BE assessment of by permission evaluation that we are further strengthening. Microdialysis based IVRT is still continuing. So then analytical method development. Now earlier we were more into IVRT, but now we are offering complete solution for analytical method development using different techniques like dissolution, liquid chromatography, RI, GCHS, LCMS, ion chromatography, all kind of analytical development and validation services we shall be providing. So this was about R&D services, but when we talk about GMP, so GMP means you have to issue certificate and you have to provide kind of analytical reports which are acceptable from a regulatory point of view, or acceptable for products which are getting released into the market. So now, since we are into GMP workspace, so we are getting our lab equipped to do QC analysis. Again, all kind of stability studies. Right now, we are not equipped with stability studies, but our SPS France facility is equipped. So some of the things which we are not equipped, we are basically referring to our different facilities, because we have a collaboration with Sortex. So not only R&D development services, now we are also giving QC analysis services, stability studies, assay, degradation product, dissolution. So all kind of analytical testing now we are offering. Then clinical and commercial batch release services we are offering, as we shall be GMP certified soon. Besides this, we are also offering support services, troubleshooting, training, consulting, audit. So these kind of services also we are offering. This is just tour of our lab facility, which inaugurated around 2 months back. Dr. Khoshia, Drug Commissioner, State Drug FDA Commissioner, he inaugurated the lab. Mr. Rolf Benz is the basically CEO of Sotex, Switzerland. So they inaugurated the lab, and this shows again lab inauguration picture, and this kind of a like USP type 4. This is for IBRT evaluation, which is automated system, French diffusion system from Logan, which is highly specialized for IVPT evaluation, LCMS, ion chromatography. So we have significantly strengthened our infrastructure to meet the need of the clients. This is our team. And this is basically our address. We are located at Ratna Business Hub. This is near on Sarkhish Babla Highway, near Sanathal sector. Now, as I mentioned, this was introduction about inception of Q3, how we originated this organization, and what kind of services we are performing. But as I discussed with Dr. Bhadia, I shall also be discussing on some technical aspects related with in vitro release testing for complex formulation, so that students can get benefited from my experience in this field. So quickly, I will just mention some of the key points here. So basically, for IVRT, it has a very important role to play when we have to establish bioequivalence of formulation. As the time is limited, I will just quickly run through with respect to IVRT as a tool for complex formulation. So IVRT challenges for complex dosage forms are many, because for formulations like liposome, microsphere, these are multiphasic formulations. So you have to distinguish between released drug and drug still in the formulation. Many of the drugs are having low solubility, and maintaining sink condition for complex dosage form is also challenges. IVRT component like microdialysis or dialysis membrane can be rate limiting during, and they reduce the sensitivity of the method. For depot injection kind of a product, long duration of studies, that makes the method development quite tricky. So these are some of the challenges that you have to consider while developing methodology for complex doses. Particularly for complex ophthalmics, IVRT is gaining more prominence because if you have to get approval for complex ophthalmic, you can get bio waiver if you can prove Q3 sameness of the complex, like injections, suspensions, or emulsions, kind of a topical ophthalmic formulations. If you show Q3 sameness, there are chances that you would get bio waiver. But if you don't show Q3 sameness, then you have to go through bioequivalence route. So, but proving bioequivalence for ophthalmic formulation is quite challenging, because you have to measure pharmacokinetics in aqueous humor, or you have to prove clinical endpoint studies. So those kind of bioequivalence are very costly and quite difficult to perform for complex ophthalmic. So people are very actively considering using Q3 approach to get bio waiver for those kind of a formulation. If you talk about ophthalmic route of drug delivery, it is a very complex route of drug delivery, because drug is distribution in several phases and complex set of condition governing release. There is a short residence time in the ocular region, and doing IVRT evaluation by conventional methodology doesn't prove that kind of the purpose. And when you talk about type 1, type 2, the media is 900 ml, 1000 or 1 liter kind of a thing. So this kind of a high excessive dilution of the formulation is not advisable for ophthalmic evaluation. Formulation temperature is also different. Here people use around 35 degrees centigrade. So for ophthalmics, regulatory requirements are different. When you talk about IVRT method for complex parental ophthalmic topicals, what are the instrumentation that are used? Standardized method are paddle method, USP type paddle apparatus 2 with enhancer cell is also there. Flow through system is also quite frequent. USP type 4 is used for characterization of complex formulation, and vertical diffusion cell, French diffusion cell is also used frequently for semi-solid and ophthalmics. So these are all called standardized methods. But every time it doesn't serve the purpose, or sometimes you have to go to non-standard method approach. And what are those non-standard methods? Like dialysis bag. People use different molecular cutoff dialysis bags. So it can be either dialysis or river dialysis mode. Then controlled temperature shaking bath is also used for evaluation of PLGA, polylactide co-glycolide based microspheres. And some other kind of a nano drug delivery. Controlled temperature shaking bath, where you incubate the sample and basically separate free drug from the bound drug, is also quite common. Then there are newer techniques like microdialysis, pulsylite microdialysis, suspension adapter, or in vitro hemodialysis. These are also becoming popular because they are serving very specific purpose for the dose response. So type 2, USP type 2 paddle apparatus you all know. So another variation of type 2 is apparatus 2 with enhancer cell, which is a smaller size vessel or mini paddle that is used for topical formulation evaluation. Like, so in an enhancer cell, there is a basically cell body, and where you can place the formulation and having a membrane on the top of it. So you can evaluate drug release from the topical formulation in a smaller volume. So that is for topical formulation is quite common. Vertical French diffusion cell is very common for evaluation of semi-solid dosage form. It is also being used for some of the complex ophthalmics. So there is a chapter USP 1724 which governs the operation methodology for ophthalmic or French vertical diffusion cell. So basically, it mentions about the type of membrane, synthetic membrane that you have to use, what kind of formulation that can be tested on by using vertical diffusion cell. USP type 4 is a very interesting equipment, because it offers solution for multiple type of dosage forms, be it microspheres, liposome, emulsion, suspensions, and beads. So it has different kind of adapters for different kind of a dosage form, particularly where the solution or the sink condition is a limitation. Drug is having very limited solubility. So for those kind of products also, it offers solution. So it is a very versatile kind of equipment. So it is comprising of a reservoir. So basically the reservoir, you keep the media, and there is a pump which pumps the media through a flow through cell. So these are the flow through cell where you keep your formulation inside the flow through cell, and the media passes through a water bath, so that it reaches to a certain temperature, passes through the flow cell, and on the top of the flow cell there is a filter which retains the undissolved particle. So what happens is the media passes through the samples contained in the flow through cell, and then after, when it emerges out of the cell, you can collect the fractions using a fraction collector. So there are different modes available: open loop, closed loop. So in a nutshell, by use of various permutation combination, it gives solution for lot of challenging problem for IVRT evaluation. Like in open loop, one, what happens? In open loop, the media from the reservoir, it goes to the pump. From pump, it goes to the cell, and then it is collected. It goes to the another reservoir, but in between you can collect the sample. So this is called open loop. So in open loop, when there is very limited solubility of the drug, you can have access to unlimited amount of media. So with very low solubility drug, it is a very good option. In closed loop, what happens is the media is recirculated back into the reservoir. So again, where you can do the sample collection by automated system. So by combination of various combination of arrangement, you have a lot of option available. Different type of cell are available for different kind of a dosage form in USB type 4. Like this is another bigger cell with 22.6 mm diameter. Conventional is 12 mm diameter. Now you can have specific kind of flow cell for powder granules, implants, ointments, suppository. It imparts overall flexibility to the use of this system. Now let's discuss some of the non-standardized systems for IVRT evaluation. This device is called floatilizer. These are basically dialysis bags. They are comprising of different kind of a molecular cutoff weights, 10 kd, 30 kd, 100 kd, 500 kd. So depending upon the molecules that you want to separate from the untrapped or bound form, you can separate the drug at macromolecular level from the other components. So this is really very good size separation or molecular weight based separation technique, wherein you can evaluate the release of the free drug from the bound portion with time. So this is a technique which is basically non-official technique, but it is being used significantly. Then shaking water bath, again, where you can incubate the sample at the desired temperature and separate the free drug from the bound drug. This technique is also used. Rotating bottle is something which is used for characterization of microsphere kind of a thing, where you can incubate the sample in the buffer and withdraw samples using rotating bottle apparatus. Disoflux I mentioned that we are doing for oral permeability evaluation, that comes under newer techniques. Here what happens, you do the dissolution in type 2, but along with the dissolution, you also evaluate the permeation of the formulation in a separate chamber, which you do concurrently along with the dissolution. So here this chamber contains a membrane which is pre-soaked in a lipid solution which mimics your intestinal membrane. So what happens is, basically it mimics physiology of intestine, whereas the drug which is having high permeation across the membrane, it passes into the reservoir, because this buffer is called permeation sink buffer. So the drugs having high intestinal permeability, they permeate to a higher extent as compared to the drugs having low intestinal permeability. So it gives you an additional tool to evaluate in vivo performance of a dosage form. That is something which is important from pharma perspective, or whenever you go to bio study for any oral formulation, it is a huge cost implication. But if before going to bioclinical studies, if you want to generate more confidence on your formulation, if you do this kind of evaluation, you will be more confident for bio success, because of you have additional data in hand. So many of the clients are utilizing this tool to generate more confidence before going to the bio study. Microdialysis, microdialysis in the past has been used for animal kind of experimentation studies, wherein this microdialysis probes were inserted inside the animal tissue, and this microdialysis probe, they consist of basically a probe having a semi-permeable membrane. So the perfusate or the media comes from the internal chamber, it goes out. When it goes out, the free drugs, it diffuses through the semi-permeable window, and so basically the perfusate, it contains only the free drug, not the bound drug. So in the past, microdialysis probe were used for animal experimentation, measuring free drug concentration in the tissue. But now what people are doing is, people are using as a in vitro release evaluation tool, because for complex formulation like ophthalmics, they want to have alternative methodology to evaluate IVRT, and the FDA is recommending quite strongly for this kind of a technique for specifically for ophthalmic product evaluation. As I mentioned, in microdialysis, what happens is you pass the perfusate through a semi-permeable probe, and the perfusate is collected in a separate container, but when this window is semi-permeable. So what happens is the free drug is able to migrate from the tissue into this, but not the bound one. So the free drug passes through and you are able to quantify free drug's level in the tissue. The similar principle has been applied for in vitro evaluation, where the perfusate or release media is passed through the probe, which is having a semi-permeable window, and the dialysate contains the free drug. So suppose this is the formulation, here, which has like emulsion or suspension, where you want to maintain the drug release level. So only the free drug will come out into this perfusate, which you can monitor. So again, an interesting tool where people are utilizing your in vitro release evaluation of complex dosage form like liposome, emulsions, or ophthalmic kind of products. Pulsatile microdialysis, where people are using this microliter syringe automated system for basically release of the media, to pass the media through the semi-permeable window of the probe. But as I told, very limited information is there in the public domain, and so people are basically curious about how this technique works. Now there are some additional things, like people have developed dialysis adapter for USB type 4, otherwise which you can reuse by using microdialysis tubing. So this is a cost-effective way of using USB type 4, because if you go for floatilizer kind of a thing, they are repeated use, they can be very costly. But if you can use something reusable, that will be real good cost effective. Then there are some suspension adapters which are compatible with USB type 2. People are using for suspension evaluation, wherein you have this adapter, you have this dialysis tube around this adapter, you fill your suspension and have it attached to type 2 apparatus, and you can evaluate suspension. So some of the innovations are going on. Again, rotating basket apparatus modified, where you can have a glass basket having a dialysis membrane attached to it for evaluation of, again, basically molecular base weight separation of complex dosage form. Just quickly, some regulatory aspects related to IVRT evaluation. There is chapter USP 714, which talks about dissolution. 724 is on French diffusion cell, vertical diffusion cell. There is chapter 1092 on development and validation of dissolution procedures. Chapter 1724, again same, it is drug release as well as solid drug product performance test. Then while you develop product for the US market, there is a dissolution method database on US FDA sites. So there you can find dissolution methods, recommended dissolution method for various generic drug products. Besides this, for complex drug products in US FDA data site website, there are product specific guidance for generic drug product development. So there you will get the characterization methodology, which are specified for some of the complex drug products at US FDA site. Pharmacopoeial form, some new chapters have come up, like drug product containing nanomaterials. There is chapter 1153. So for the researcher, for the students, they should refer these two, because a lot of new techniques have been mentioned here for characterization of complex dosage form. So these two would be really for of good interest. Chapter 1153 and 1001. 1001 deals with in vitro release test method for parental drug probation. So definitely the students find these new chapters or these new pharmacopoeial forum chapters interesting. Again, this shows how dissolution method database specifies a methodology for different kind of a formulation. Like this is injectable suspension. So if you go to dissolution database, it will tell you which apparatus you have to use, which medium, and when it was updated. So this is just to show you how you can get relevant information from method database for complex injectable drug products. This is for B specific guidance for generic product, like for propofol. What guidance would say, what kind of a Q3 characterization you have to do, along with Q3, whether you need to have IVRT method or population bioequivalence of size population, you have to do it. Now, if you do all three in vitro option, then you will get bio waiver. So like for propofol injection, either in vitro option or in vivo option. So if you are able to do, no need to do bio study because you will get bio waiver. So this is something very interesting from the industrial point of view, because they can save a lot of cost if they can do Q3 characterization. Interesting to see that for ophthalmic drug products, there is no specified method listed in database. So what it says, develop a method. So there is a lot of gap when it comes to characterization of ophthalmic drug product. It shows the regulatory landscape is still hazy. The complex ophthalmic drug product, again, Q3 characterization and in vivo option. So when you go product specific guidance for ophthalmics, again you will find here what kind of a in vitro options that are available or what are in vivo options. So companies are trying to move towards in vitro option, including Q3 characterization for getting bio waiver, so that they can enter into the market into a cost effective way. And this is where the speed matters. How quickly you can meet regulatory expectation and how quickly you can get entry into the market. And activities of our organization is basically catering to those kind of a Q3 characterization needs. With this, I have come to end of this talk. I hope you have find it to your interest. Kindly, if there are any question, it will be a pleasure to answer.
Thank you, sir. Your talk was really very enlightening and it was really very knowledgeable, full of knowledge, and our students surely have taken a good take-home message from your talk. And really, it is very interesting about all the services, like both R&D as well as GMP services which your company is providing, and the way different type of in vitro release testing that are developed for the complex formulation. So you have really nicely explained through the example of the ophthalmic delivery systems which you have delivered right now through your, and the students are really motivated. I think they will also try to develop some novel formulations and may come to you to take your help regarding the dissolution parameters, to develop some kind of novel IBRT method to study its release characteristics. Also, sir, very interesting thing I got from your talk, one of the services which you are providing is reverse engineering, and you have really very well reverse engineered the name Vitro to Ortiv. Really, it's a very good idea, and I really like the name of the company, Ortiv Q3. It's a very good name.
Right.
So I think, sir, there is no questions, I guess. I don't think anyone have any questions. You can post in the chat box.
Madam?
Yes, sir.
I have one question.
Yeah, sir, sure.
Mukesh, sir?
Yes, sir.
Yeah, thank you very much for a very informative session. Sir, I have been working on developing therapeutic contact lens. Okay. So any idea regarding developing a dissolution method for such thing? Because we have been doing its dissolution with some of the published literature, whereby I used to take just 2 ml of the dissolution medium, trying to mimic the in vivo condition. And at the time of withdrawal, we generally withdraw entire 2 ml and replace it with the complete 2 ml of the simulated tear fluid. Okay. So have you come across any product development or any project whereby you have tried to develop some dissolution method for therapeutic contact lens?
See, when you talk about contact lens, are they impregnated with some kind of a drug or what?
Yes. We try to incorporate a drug into it. Okay. We incorporate by direct drug loading. We may mix it with the ink with which it would be ink would be printed onto the contact lens. Or we try to incorporate drug via formulating microemulsion or nanoparticle. Yeah, so that is really an interesting concept. So since this is a new kind of a development, so the idea, for ophthalmic drug products, regulatory bodies have certain expectations. Like it should be a little bit physiologically relevant. You should not use very excessive diluent.
And since residence time in the eye is less, so they want more frequent sampling. And it also depends what kind of a duration of action you want from your, you are expecting from your device. If it is a say 2-3 hour release, or if it is a long acting kind of a stunt. So based on intended duration of action, and based on the kind of the release media that as you rightly use 2 ml or less kind of a media. So that approach that you are following is right, but you need to take into consideration the intended duration of action.
Yes.
Right. And based on that, since this is a new and there is no regulatory kind of a method available, so whatever you do, just try to be as close as possible to the physiological relevant condition, right? And all you have to prove is there are some interesting FDA presentations. You know, if you go to the internet and see for ophthalmic drug product, what are the FDA expectation, you will find some FDA presentation on their expectation with respect to ophthalmic formulations. So there you will get more insight. What are the regulatory
Yes.
expectations, right? So in the nutshell, you are going into the right direction.
Yeah. Thank you, sir.
Thank you.
I think Savle sir also wants to say something.
Yeah, I just wanted to, this is not a technical question, but as this is an inspirational talk, Dr. Mukesh, you want to know what triggered you to kind of quit very successful professional life at industry and leaving employment and becoming an employer?
Right. You know, when I came here in Ahmedabad after Wokhad, so I was working with my past company, Dorizo. So, you know, after acquiring so much of, I was having around 25, 26 years of experience. And the kind of work I was doing in my past company, it was some kind of a monotonous. So, and you know, you need to have the right kind of a people. And when we were discussing in our friend circle, we have this kind of experience, but somehow is there any need which we can address? So we got the answer, yes, there is a need and we can address that. So the problem was there and the solution was looking visible. So that prompted us to take the plunge. It was a risky decision, but but otherwise it would have been too late, either now or never. So somewhere you have to take the plunge. So we took that.
Yeah, I think as a startup, you have to have that risk-taking
Right.
capability. So,
Right.
but I think very impressive. Within 2 years, you could attract measures like SOTAX to show interest in your business. And you had, I mean, growth at the rate of 200% increase in your kind of revenue, as well as I see global clientele getting attracted to you. Congratulations for the excellent progress.
Thank you for your encouraging words.
Yes, Praful sir also is there with us, so he also wants to say something.
Okay, Mukesh ji, namaskar.
Good afternoon, Dr. Saab. How are you?
Sorry, I was traveling, so could not connect with you at the start of the session.
Thank you.
But
Thank you.
it was very interesting. I joined in the between, a very nice technical talk. And as Savla sir has suggested, like, you know, being an innovator now and an entrepreneur, we look forward to a lot of inspiration from you for the students, correct? So maybe a few of the students who wish could visit your lab, research lab, and see how the whole thing has you have developed it over there. Is it possible?
Yes. So if you want to have a student visit, so that is not an issue. Anytime they can just work out as per convenience, because right now qualification and all is going on. So so to make a lab or functional.
Okay. Hello.
Hello, madam.
Yes, sir.
We are not getting.
Yes, sir. I think there is some issue
Hello.
with Mukesh, sir.
Madam, am I audible? Just,
Sir, we are not able to hear you. Yeah.
Dr. Bhadia, are you able to hear me?
Voice is breaking a bit.
Okay.
Connectivity issues.
Now I think it is okay.
Dr. Bhadia, what I was saying is, of course, it will be a pleasure to show our lab to the students.
Okay.
But, as our qualification and all the things are going on, so
Right.
Definitely, it will be
Okay, we'll work it out. Not an issue. As soon as things are
Sure.
ready with you on the side, we'll try to work it out. Not an issue.
Sure.
Okay.
Sure.
Yeah. Thank you so much for spending your time and being with us today, Mukesh ji.
Thank you.
Yeah.
Thank you.
Radhika madam,
You're on
your
mute.
voice is.
You are on mute, Dr. Radhika.
Yes, sir. So, sir, thank you very much. And sir, as a token of love and appreciation, I would like to present a certificate of appreciation to you. So, Mukesh, sir, can you please share it?
Okay, is it
Not you, our another Mukesh sir is there.
Okay.
So he will be presenting the certificate.
Okay.
So, Mukesh sir, Mukesh Kher sir, please share it.
Maybe in the meantime, one quick comment is LMCP has MOU with RTV Q3. So we have collaborated. So we can do a lot of such initiatives collectively. This is good industry academia partnership as well.
Yes,
So
sir.
I wanted to bring to everyone's notice. Yeah. Go ahead.
Particularly this Atal Incubation Center, I think it is doing a fabulous job of having very niche kind of infrastructure. So it is really very nice that we have a collaboration with Atal Incubation Center also. And in future, we shall definitely explore your facilities because we are not presently equipped with some of the things which you are having. So definitely in future, we shall explore using those kind of facilities.
Sure.
So, sir, I request our principal, sir, to present the certificate of appreciation virtually on this platform.
Mukesh ji, please accept our token of appreciation and gratitude. That you spare your valuable time, and definitely your talk will enlighten or enhance the knowledge and motivate the students specifically.
Thank you very much, Dr. Thank you very much for kind gesture.
Thank you very much.
Thank you, sir. And now, before we end, I would like to invite Dr. Ketan Ran, sir, Associate Professor, Department of Pharmaceutics, to deliver vote of thanks. Sir.
Thank you, Dr. Radhika. Let me extend a vote of thanks on behalf of LM College of Pharmacy, LMCP, Institution Innovation Council, AIC, and SSIP cell and committees of LM College of Pharmacy. We had absolutely dignified innovator, entrepreneur, and scientist as this one, who has summoned a graceful, befitting, and not to the year 2021. Dear participants, India has embarked a new elevation in innovation and startup since few years because of innovator like Dr. Mukesh sir. Sir, we are really honored and appreciate you for your inspired presence among us at LMCP platform, and also for delivering a very wonderful, thought-provoking, inspiring, and very informative lecture focusing on aspects of creativity and innovation through the technical talks of pharmaceutical science research, as well as your company's growth story with 200% in such a short span of time, with all its offerings, services, and technology at Ortiv Q3 Research Private Limited. So we at LM College of Pharmacy are very fortunate to have you with us in today's webinar series on My Story. Thank you, sir, for accepting our invitation and delivering your expertise and experience in this lecture series on My Story. Your lecture has truly inspired our student to excel their career by being creative and innovative, and to become a job seeker, to become a job giver rather than job seeker. At LMCP, we could do such things in a better way because of our strong support from Principal Dr. M.T. Chabaria, sir, who has been working with his all intellect and supporting us to keep us on the go. So thank you very much, Chabaria, sir, for raising the level of merit and intricacy at LM College of Pharmacy. We are thankful to Professor Praful Baradia, sir, IIC President, AIC CEO, Dr. Srinivas Savle, sir, and IIC convener Dr. Radhika Pandya at LMCP for taking their efforts and facilitating this webinar to bring such an expert on LMCP platform to deliver lecture in this webinar series of My Story. So thank you all of you for your encouragement, guidance, and support. We are also thankful to all members of IIC, AIC, and SSIP that we could not have done this webinar without your commitment and untiring efforts beyond the call of duty. I am also thankful and grateful to all the staff members of LM College of Pharmacy because this would only be possible with their relentless support. We also thank all the participants and students for being part of this webinar series. We know we cannot thank everyone enough for their involvement and willingness they have shown to take on tasks beyond their comfort zone to organize such webinar series on a regular interval for our students. So Mukesh sir, we have very talented young minds who are full of innovative and creative ideas. So Savle sir has rightly pointed out that we have a collaborative work. So sir, we come out with some innovative project, especially on formulation. So we'll be looking forward for research collaboration with you, as well as for placement of our student with your organization. So once again, we want you to know that we are deeply appreciative of the support, Mukesh sir, with AIC as well as LMCP. Thank you very much. Over to you, Radhika madam.
So thank you again very much, sir. Really, it was a very good session, and I hope that in future also we will keep communicating with you and collaborate with you. And sir, before we end the session, I would like everyone to turn on their video so that we can capture some photos. So request all the participants to please turn on your camera. Okay, sir. So, sir, thank you very much.
And thank you very much. And it was really an honor to present our journey, our inception of our organization. I really enjoyed the whole session, and I really felt very happy to show such kind of a response from the LML college and the faculty members. So really, it was a pleasure to interact with you.
Sir, it is our honor also. Thank you, sir. Okay, sir. So we will just end.
Thank you, Dr.
Thank you. Thank you.
Thank you. Okay.